I try to imagine a fellow smarter than myself, and then I try to think, what would he do?
Charge up your axons, ready your receptors, and shift your lobes into upper beta phase. You are listening to Smart Drug Smarts, the podcast dedicated to helping you optimize your brain with the latest breakthroughs in neuroscience, nootropics, and psychopharmacology.
Hello and welcome to Smart Drug Smarts. I am your host, Jesse Lawler, excited to bring you the 8th episode. Yes, the 8th episode of this podcast, and the first ever episode that we've had that actually I think is going to come out within a week of the previous episode, which is a major milestone because this has always been meant to be a weekly show, and it has not been until this point. But I think we're over that hump. I think it's going to be weekly from here on out, and if you've been bearing with me since the beginning, I really appreciate your sticking in there as I stumble my way through this. And in fact, there's one major stumble I've got to pre-admit to this week, which is that I made the mistake of trying to record a conversation with my guest over a cell phone network rather than doing it on Skype, and it was really pretty crappy audio quality on his end, and I should have really aborted the thing midway through and just waited until we could get a better connection, but I didn't, and now I'm kind of kicking myself that I didn't. So anyway, the audio is not going to be what it should for this episode, but bear with me. We'll not make that mistake again in the future, and the interview is well worth listening to. It's a super smart dude named Stephen Bourne. He is a postdoctorate at MIT and is currently working in drug production through continuous flow processes, which, if that sounds technical and awesome, it's true in both cases. He'll tell us about that. He'll tell us also about the drug sulbutiamine or sulbuthiamine, or, well, he'll also pronounce it for us, which I cannot seem to remember the correct pronunciation of the darn thing, but it's a pretty well-known smart drug that's been around for a while, invented by the Japanese. Also, if you hang around for the ruthless listener retention gimmick, I will tell you about my own experiences with piracetam, which I've been taking for coming up on 3 weeks now, and my shocking results. But first, before we get to all that, let's do This Week in Neuroscience.
Smart Drug Smarts, This Week in Neuroscience.
So the Pharmaceutical Journal out of the UK asks, what's the situation with smart drugs use among UK university students? And I wouldn't call it a fear-mongering article, but there was a little bit of a tinge of, well, we know there's some crazy stuff going on in the universities over in the United States of America, but has any of this madness made it over to the British Islands? And based on the statistics in the article, I would kind of say the answer is no. They interviewed a bunch of students, and only 8% of them had heard the term smart drugs. And when smart drugs was sort of given as a basic definition of what people are talking about when they use that term, this number jumped up to 24% that said, yeah, I've heard of things like that. I know that they're available out there, but only 6% of the students admitted to having used a smart drug to help them study, and, well, 46% said that they would consider using a smart drug. And like any journalist, there's a little element of looking for the salaciousness within the story that he's telling, and in this case, it's 46% of students might be willing to take a prescription drug without a prescription, or head for the hills, the sky is falling, and needless to say, I just can't take that seriously. Most university students are binge drinking on a regular or semi-regular basis, doing things that we know are rotten for the body and the brain. So being overly concerned about the small percent that is taking smart drugs and the fact that there could be some long-term, unknown, as yet undiscovered negative consequences, but still, I'd say in comparison versus other things which are far more common in society, it's just kind of looking for the touch of gray on the overall silver lining that is neural supplement. But this is just me rambling at this point. Let's move along to the main interview. This is Stephen Bourne of MIT.
Smart Drug Smarts.
Thank you very much for joining me. I guess, first of all, can you tell me just a little bit about your background in chemistry and biochemistry and neuroscience? I mean, you've obviously got a huge wealth of academic credentials to talk about.
I grew up in San Francisco, and I did my undergrad at UC Santa Cruz, and I couldn't decide which between majoring in chemistry or biology or biochemistry. It all sounded so cool, but after I took an organic chemistry lab for the first time and got to feel a real connection to the science that I was doing by, at the end of the day, you can actually hold it in your hand.
Right.
I definitely decided to do that, but I didn't want to rule out biochemistry or a lot of the molecular biology because the organic chemistry I was learning has direct implications on biology, and so I wanted to find out just what it was doing. So I ended up double majoring, and then I went off to grad school and got my PhD in synthetic organic chemistry, making all those molecules that nature makes so easily from scratch. and it takes years, but you get to better appreciate what nature does and making what nature's so good at.
Yeah.
So after postdocs at Berkeley and now I'm at MIT, I'm currently learning how to make pharmaceutical drugs or specialty chemicals in continuous flow, so in a pipe rather than in a flask.
Cool. Well, we were going to talk about, smart drugs in general and maybe sulbutiamine. Am I pronouncing that right? Is it sulbutiamine?
Sulbutiamine?
Sulbutiamine.
Whatever works. I don't even want to look at it.
Well, I saw that basically it's a synthetic derivative of thiamine, the chemical that I guess you're sort of a normal thing that we try to get enough of in our food, and that there's a U.S. recommended daily allowance of and stuff like that. So I thought maybe it was going to be pronounced the same way, but you never know with these science words.
Yeah, no, it's just, I looked at it and I didn't look up how it was made. I can tell how it was made, and actually I thought it was a great idea how they made it. So thiamine, vitamin B1, is a very useful vitamin to have. I mean, it's a vitamin, so you need to, people realize that they were lacking it based on some sailors in Asia, Japanese guys, found out because they had a rice-based diet and didn't eat enough veggies or meat or anything, and while they were out on their sea-conquering voyages, back I think it was around the World War I era, was they'd get these physical debilitating effects that later became characterized as the disease called beriberi. And they linked that to thiamine, some of the consequences of vitamin deficiency in all their different kinds.
Yeah, this is kind of like the problem that they had with scurvy in the 15th century, only 4 centuries later.
Yeah, like vitamin C for that one.
Yeah.
Exactly. Vitamin D, very important. I think the Japanese were a little bit more industrial in trying to kind of resolve this remedy. So I believe they're the ones who developed this drug, sulbutiamine, realizing that thiamine passes through the body very easily, because it's a salt. So your kidneys just process it like it was salt, like it just flies right through. And so the trick was that how do you kind of keep soldiers healthy with the open sea for a long time without actually fresh veggies or meat or anything like that, or just like on rice or on a minimal diet. So they went and they played around with thiamine. I mean, it's very polar. We've got this thing called a blood-brain barrier, which keeps most drugs out of our brain. It's kind of like a moat around your brain, which protects you over time from eating the wrong thing and then, dying. So it's important to have a blood-brain barrier. So in order to get a drug up into your brain, and in this case, it's believed that thiamine does have some distinct beneficial effects on memory and other things. The idea that it goes to the brain was based on a lot of the physical symptoms of beriberi and a lot of this vitamin B1 deficiency, which, if you look it up on Wikipedia, pretty much you just start acting really slow and your peripheral nervous system isn't firing as much, and it gets worse from there out. But the problem is, of course, is that you can't really go into anybody's brain and monitor this real time because later the patient would probably die. But they've done, but they know it gets to the brain because they've modified thiamine to be greasy and nonpolar, or at least nonpolar relative to thiamine. So from going from this very polar charged species, which the blood-brain barrier can filter out very nicely, things that are more nonpolar can get through quite easily. And this tactic has been known for hundreds of years in drug synthesis. I mean, they knew this back when they were converting opium or morphine to heroin. Basically, you're making it more nonpolar, so it goes through the blood-brain barrier quickly. And so that was the goal, what they were trying to do here. They wanted to make a vitamin B1 that lasted longer. And so they came up with this way where if you were to look up vitamin B1 on Wikipedia, you would see that in the molecule there is a sulfur atom. It's in a ring that is charged. If you had an oxidant, something like chlorine or, chlorine's a good example. What happens is the sulfur gets oxidized, and then if you have anything around like solvent that would react with that sulfur, that oxidized sulfur, then you get the ring to open, and the molecule would no longer be charged. So people who were, the first two drugs, they would pretty much have like methanol attached to thiamine. And so people, after a while thought, we could put on all these things to open up this ring so we can make it nonpolar so we'd get through the blood-brain barrier. But for every molecule of vitamin, we're giving them a molecule of methanol or something like that.
Right.
Methanol gets oxidized. That's not good. Someone had the bright idea. If we do this, if we oxidize sulbutiamine in the presence of nothing, or at least an inert solvent that won't interact with it, then one molecule of thiamine would be oxidized, and another molecule of thiamine would react with it. Now you've got a dimer of thiamine, which is great. And that way, one half of the molecule is thiamine, and the other half is thiamine, for the most part, opened up. And then they isolated it just so it could be more.
If I can throw us off course for a quick question, just thinking about the blood-brain barrier, and obviously it makes sense that we would want to protect our brain possibly more than any other organ, because it's pretty much the most important organ we got. But how come we have a specific blood-brain barrier around that organ and many of our other ridiculously important organs that might be able to sustain a similar barrier don't have one?
That's a good question. So I would say that It's mostly to keep bacteria out. I think that our immune system is pretty good when it comes to the rest of our body. If you've got like some sort of infection somewhere, usually your body can address it. Not a pleasant thing, but it's not affecting the control system. So I think that we've developed this over time because of not just bacteria, but also perhaps when we're foraging for food and we're like, can we eat this? If it's like a potent kind of plant or something like that, some things may kill less developed creatures, but not us. Might make us really sick and throw it up. But generally, the blood-brain barrier is kind of developed as a needed barrier to keep us functioning.
Cool. So back towards sulbutiamine, I feel like I'm still saying that wrong. And obviously there's been a lot of studies that have shown there's beneficial effects, working memory and speed of processing and some of that stuff. Can you tell us something about that?
Yeah, so this brings us back to the problem that people have with developing tests to figure out problems with the brain or diseases with the brain. Alzheimer's is a good one, where there's a great need for an Alzheimer's drug or a therapeutic that would help alleviate those symptoms. But one of the problems are, because there's such a high degree of need for it, that any successful drug that comes out for it would be taken by a huge number of people. So it has to be intrinsically safe. It can't make the person worse off. So there are certain levels of criteria that must be met. One of them is that, you have to know what the hell's going on. Or at the very least, you have to meet your clinical endpoints, which mean that what you say it does, it actually does. So a lot of people have trouble trying to apply the scientific method to Alzheimer's treatments because I mentioned so far about trying to make the drug greasier to get into the brain via passing through the blood-brain barrier. But in terms of what's actually going on at the receptor level, is very difficult. We're getting better with our ability to see into people's brains while they're living and continue to stay living. So one way that we're able to do that is by radio labeling. So we can actually swap out one of the items in a drug for something like a radioactive thing, nothing that you need to worry about long term, but something that shows up in a CAT scan or an MRI. And you can see like where it might collect certain sections of the brain. And because we know so much about certain sections of the brain, like we can tell that, it's affecting this, and the fact that it's staying there for this amount of time, given the fluid dynamics of the brain and recirculation, what might be going on. But in terms of the actual receptors, it's a little difficult. It's hard to work on human brains that are living, but if you can work on mice or some other smaller organism in a way that hopefully makes them pain-free, and they do that because they care after them, they'll try to do stimulus response, or better yet, just performance tests are a very easy one. And so when people do all these studies, they try to do the least invasive test they possibly can. Some of the more invasive ones, they'll actually do it like just nerve cells, cultured nerve cells, just to see like what happens with that just on the cellular level. And that actually, I think, does a pretty good job with being able to study action potentials in cells, where you can actually put on a little patch clamp, which you can kind of think of it as a jumper cable for a nerve cell. You need a really good microscope, and you need to be really patient. A lot of fine motor control to get it on there, but you're actually able to measure the nerve signal going through a synapse or through a nerve cell.
When you say fine motor control, is there actually a human operator involved in attaching something to a single nerve cell? Is that not done by some super high-tech newfangled computer?
No, I think it's done well. Basically, there's an electroneurophysiologist, and pretty much you've got calipers. Like you've got dials that you can turn that turn other dials that give you the fine motor control necessary in order to get it just right there. And these things are highly sensitive enough to be able to tell the differences in microvolts or whatever potential is, I forget, across a membrane. And so you can actually trigger a depolarization event where if you've got like an ion channel that you're looking at, if you've got a difference in salt concentration across the membrane, you put in the signal, it will open up, the membrane will depolarize, and this can actually induce a electrical charge or a nerve signal as it propagates down the nerve cell. In this particular case, for this drug, when you kind of read through it all, the way that sulbutiamine works, I believe, it acts through a variety of ways, but I think what it does is it just increases the potential of things. So basically it makes you sharper, but it doesn't improve your long-term memory as much as your ability for events to more ingrain themselves in your consciousness.
It seems like that's sort of a common theme with a lot of smart drugs. Like you said, we know that they have an effect, that they do something, but the exact mechanism by which they work is sometimes still a gray area.
Yeah, actually, that brings us to the present, where a very real debate right now is whether the way that we go kind of investigating drugs in a targeted fashion towards distinct biomolecular targets is that useful relative to treating everything like a black box like what it was like 50 years ago. a lot of people will credit 50 years ago, there was a lot of low-hanging fruit. I think that back then, no one really knew what was going on at the biochemical level, and as much as they knew that it worked and it had low symptoms or low side effects. And there are ways that you could manipulate a molecule in order to maybe increase its potency and decreasing the side effects, which is kind of what the whole drug industry wants, better, safer things. But ever since, we solved the code of the human genome and realized that, unfortunately, I think wheat has more genes than we do. What makes us more complex organisms than wheat is that we're very highly regulated. So we have less gene products, but there are more feedback loops and control loops. So we have more regulated biochemical mechanisms, and so this leads to greater complexity than something as simple as gene product A gives protein or enzyme product B, and that's just the way it is. So because there's so much homeostasis going on at the biochemical level, we've, and couple that to the Human Genome Project, so a lot of people treated it like the mad rush to patenting all the DNA, and I laugh now in retrospect. But yeah, you can patent all the DNA you want, but the more we learned about our biochemistry at the biochemical level, the more we realized how complex we were.
Yeah, it's like we were expecting to find a whole lot of nouns, and what we found in addition to nouns were a whole lot of verbs and modifiers and adjectives, and we're like, oh, didn't really see that coming.
Yeah, and then we realized that there were like 50 billion different languages.
Yeah.
It made things very complex. Now, there were certainly some biochemical targets that over the last 10, 15 years have really stood out in terms of being good models of, look, we actually applied this reasoning to some, kind of rational drug design, and we believed that we could model using some 3D software that a drug of this design would fit into the active site of this enzyme to kind of gum up the works to keep this enzyme from misbehaving. But again, the problem is that as far as drug design goes, we are a very complex, highly regulated species. And if sometimes one biochemical pathway is shut down, there's a feedback loop, and so your body can work around it. And so when it comes to drug design, it makes things a little trickier. And so I think that we had a lot of success in the past because we would screen for, one could say, a phenotypic effect. Does this chemical actually improve the condition of X? And people didn't really need to know how well it worked, so much that it worked. And actually the FDA still, people moved away from that model of looking for drugs because it was too uncertain. They like certainty, they like predictability. So we've got really great analytics for understanding everything from just X-ray crystallography to data mining the genome to try and figure out, or you can actually, they've gone beyond genome now. They've actually said, okay, we know enough about DNA to know what proteins it codes for. We've got the proteome. All right, what about all the fats that code for, all the different fats? It's a buzzword, but there's so much that we're learning, that we're continuing to learn, and it's just basically one big, complex, crazy place. So sometimes it's better to treat it like a black box and just kind of look around for what works and what doesn't, because ultimately you have to scale, up the clinical trials to work beyond the biochemical level, right?
Right, and when you're going back to analyzing the phenotypic effects, which is sort of where they started from.
Yeah, exactly. And the problem is that you miss a lot in between. And more importantly, what you're starting from, at least in the modern version where, let's say you're going to be looking at a drug that, looks at a cancer cell, it's easy to make drugs that kill cancer cells. The problem is that cancer is not a cell, it's a tissue by the time you're trying to treat it with drugs. And so how do you get it through the tissue? Because cancer cells develop sometimes these very dense outer tissue coverings. So how do you get it through the tissue, or how do you get it delivered to the cancer in the first place? These are really basic questions. How do you get a message to someone if there's no mailman? But that's the problem, again, with any kind of drug that affects the brain, is that there's a whole lot we don't know. And I think that's one of the things that I think is great about the blood-brain barrier in some regards, is that we treat it like a black box. And so, anything that develops from it, you can actually do some of these tests to figure out what's going on and still be able to get some information out of it without ever having to, be beholden to actually cutting open people's brain.
One question I've got for you, you're obviously a very, very smart guy. What's sort of on your mental checklist for keeping your brain as sharp as possible?
I'm actually not that smart in the grand scheme of things.
Oh, phooey.
I have the academic checklist of where I've been and what I've done, but most of that was done through hard work and beating my head against a wall. I was never one of those gifted students who everything came naturally to them. I've got, my family is a family of foodies from San Francisco. I love how to cook. That's, I think, in part why I took the organic chemistry so well, is that it's almost like a recipe. It's how do you caramelize sugar? Well, sometimes purifying chemicals is the same way. How do you distill vodka or, a perfume? It's not too far from each other. A lot of the ways that we prepare food are the same ways that you can do chemistry. So, at the end of the day, being able to, make a pot of really strong tea and being able to extract the caffeine from tea leaf and then recrystallizing it and then holding that caffeine in my hand at the end of the day, I felt a good affinity to that. So.
Your modesty is impressive, but we're going to put some of your credentials on the post that'll go along with this podcast interview, and people can make their own decisions as to how educated you are or aren't.
So actually, I learned this while I was an undergrad, when I needed my brain the most. I think a lot of people who listen to this podcast might kind of be around that age group where if they are in college and learning all about the wide world and they want to know what works for the brain. Two things. One, a stable supply of blood sugar. So eat food, good food, healthy food, preferably something you cook from scratch. So one, throughout the day, you need good blood sugar, because if your blood sugar goes down and you're just feeling famished, you're not thinking about what you're doing. You're thinking about what you should be eating.
Mm-hmm.
So that's number one. My days were spent kind of figuring out what I was going to eat and how I was going to eat and try to fit it in between classes and everything like that. Bring a small lunch. That's half of it. The other half of it, get up early in the morning. I know it sounds badly to undergrads, and I can only remember what it was like. And I remember it. The freedom was great, and early classes sucked. But did a study. I was taking this one hard class and had to write all the papers out. And oftentimes it was me working till dawn. And I could actually count the progress that I made. And as a general rule, general trend, the efficiency of your brain to actually do, I would say, good work in terms of writing, reading, or doing anything kind of scientific, or this even held true for lit courses. After 10 p.m., that efficiency was down by half. So for every hour you're putting in, you're only getting in a half hour of actual work because your brain is tired. So if you can actually change your circadian rhythm to actually a circadian rhythm, and when the sun comes up, you should start think about waking up. And then when the sun goes down, you should think about getting in bed or getting home, where you don't have to think. And really, that's just resting your brain, because it works an awfully hard time, especially when you're an undergrad in college and that needs to rest. And so, I think that's the two things really that a lot of undergrads lack are nutrition and proper sleep. And conversely, I think building a body that is robust enough to deal with some of the abuse that we dish out. If you can take up some aerobic exercise a few times during the week, doesn't really matter what kind. That way, if your body's in a healthy state, it can keep you focused on task. It actually increases all these good things that people talk about, like your attention. I mean, I ran a lot. I was never any good at running, but I ran, and I never get sick, and I've got this fiendish sense of work ethic. I'm just lucky because I actually like what I do. So find something to do that doesn't seem like work your ass off. Just don't let anybody take advantage of you. That's what grad school's for.
Nice.
Yeah. There's actually a particularly good book. I'll recommend it. You can probably find it used if you're interested in drug design and drug action. It's book's called The Organic Chemistry of Drug Design and Drug Action.
Appropriately titled.
It's by a guy named Richard Silverman. I had this, God knows how long ago. It's an old title. It's just publication date 1992. But it's easy to find cheap copies, and it's not too thick a book. And it actually talks about all the things that they look for when they think a drug from a pharmaceutical company perspective, where they actually have to screen against toxicity. What will this do to your body? What will this do to your liver? Will this react to your liver? They look at all these things, but they put so much money into looking for that. And then, the FDA requires that too, because they don't want us selling crap.
Sure.
So, but this is actually broken down in terms of what it is that they're looking for. See how long it's used in your body, how long does the activity last, and how could it have toxic side effects, what kind of things do they look for in the molecule. And it seems that you've at least flipped through an organic chemistry book before, but if you have, then I recommend the book.
Smart Drug Smarts, the podcast so smart, we have smart in our title twice.
Thank you very, very much to Stephen Bourne for joining us on the episode. Super, super smart guy, a wealth of knowledge, obviously, and I think we'll try to get him back in the future in a better audio circumstance. As for next week's episode, I have reason to believe, if I look into the future, that we're actually going to have a futurist on the show talking about what we can foresee for the future of humanity in general and the human mind and sort of cognitive performance in specific. But anyway, more on that soon. I guess we'll find out next week. Just wait a week and we'll get the answer to that one. As for what I promised you at the beginning of the episode,
Smart Drug Smarts, ruthless listener retention gimmick.
I promised you back at the beginning of the episode I would let you know about my own experiences taking piracetam over the past 3 weeks. Piracetam is one of the racetam family of smart drugs. It's probably the oldest and most well-known and well-studied. It's been around for a long time. It's something that's often given to seniors that are starting to suffer from pre-Alzheimer's or senior dementia. But it's something that's also been shown to have significant neurological benefits for people who are younger. So I figured, heck, I'm going to start taking it. I know some people that are my age and younger who are taking it, and the results are in. Drum roll, please. I have felt absolutely nothing. Absolutely nothing. There's no placebo effect going on here because I just could not for the life of me tell whether I took the stuff or not. Might as well be a big glass of water or something like that. So anyway, what does this mean? It might mean that I'm taking the wrong dosage for myself. 1,600 milligrams seems like about a middle of the road dose. Some people take less. Some people, I think it's been shown that you can take up to 9.6 grams a day without any sort of negative side effects. I'm going to continue to experiment, do a little bit more reading, and I think I'm also going to look at a piracetam episode sometime in the near future. Anyway, that's all that. Okay, so that is the entire episode now. If you liked what you heard, please recommend this podcast to your friends. Leave us a review on iTunes. Do any of that great stuff. Pass the links around. The show notes for this episode will be online at www.smartdrugsmarts.com, which, if you're to believe rumors, is soon to have a little overall look upgrade to the website itself. We'll be back next week at the same time, same podcast, and with the same unflagging commitment to helping you fine-tune the performance of your own brain. Have a great week and talk to you soon.
You've been listening to the Smart Drug Smarts podcast. Visit us online at www.smartdrugsmarts.com and subscribe to our mailing list to keep your neurons buzzing with the latest in brain optimization.